Okay, levels of DNA compaction in what type of organism?
Us.
Eukaryotes. Okay. So in eukaryotes, first level is nucleosome, which we have talked about. We've done experiments with micrococconucleosigns. Okay. This is the winding of the DNA about 200 bases. It's not always 200, but it's roughly 200. Around that octamer of histones producing what is also called the beads on a string, okay, pearl necklace, or 10 nanometer fiber, which is the diameter of the fiber. Okay. Then this DNA, or beads on a string, is going to be, the H1 histones are going to interact with each other, making what's called a solenoid structure. Okay. It's basically now going from the beads on a string to a more cylindrical structure, like a cylinder. Okay. That is also called the 30 nanometer fiber. This is the diameter of that fiber now. Okay. The last level of compaction is radial looping into pretty flower structures, rosettes, as well as scaffolding, which means the rosettes are going to be stacked on top of each other. Okay. Okay, so nucleosomes. You should already know this. So there's an octamer of 4 histones, 2 H2As, 2 H2Bs, 2 H3s, and 2 H4s. Around each octamer of histones, okay, so here's an octamer of histones, about 160 bases of DNA are wrapped around. It's about 1.5 times, okay, around the histones. Then there is a linker region, which is roughly 40 base pairs in length. This is where H1 is going to bind, which is not part of that octamer. Okay. So here's the beads on a string in an electron microscope image. DNA does not coil smoothly. The sequence of the DNA actually dictates where those nucleosomes are going to be formed. Okay. And then the spacing and structure affect the genetic function. So why would you want the spacing more spaced out? Why would you want less nucleosomes in an area?
Divide.
Less. Less. Yep. Gotta have that DNA accessible. Less nucleosomes makes the DNA more naked, less clothing on it. Okay. That will allow it to be more accessible to the transcription machinery. More able to be transcribed. If those nucleosomes, there are more of them in an area, they're less accessible, they're transcriptionally inactive. No transcription. Does that make sense? This is more of a molecular view looking at those core histones. So the blue is the DNA wrapping around the core histones about 1.5 times. And no, your histones don't really look like Trix cereal. But there's 2, let's see, these are 2 H2As, 2 H2Bs. Here's an H4, and there's another one on the other side, H3, and another one on the other side. So there's a lot of protein-protein interactions, but there's also a lot of protein-DNA interactions here. Okay, second level of compaction is that solenoid structure, okay, making the 30 nanometer fiber. Okay, so what happens is that each of these nucleosomes, there's that linker region where H1 is bound. Now the H1 histones are going to start interacting. And they are going to start making the nucleosomes into a nice ordered like staircase structure, a solenoid structure. Okay. And the diameter of that solenoid structure is 30 nanometers. Which here you can see the solenoid structure down there. Here's kind of a top view where all those H1 histones are interacting. Just to let you know, not to throw a wrench in it, this solenoid model is on the out. New data is showing that it's really now a zigzag model. We're going to stick with the solenoid model for the moment because that's probably what I'm going to test you on. But genetics is a very new field. Solenoid model seems not to be the most accurate model. Okay, so chromatin meaning DNA and protein, okay, is going to exist in this 30 nanometer fiber or a more condensed state, okay, during interphase, but it's going to be maximally condensed during metaphase of mitosis or metaphase of meiosis. Why? Why should it be more condensed during metaphase of mitosis or meiosis? What goes on in metaphase? They're lined up. What happens right after they line up? They get separated. What if they weren't condensed? What if they're all squiggly and all interacting with each other?
It'd be hard to separate.
You wouldn't get the good separation. You get lots of chromosome breakage, pieces of DNA being ripped off. Okay? So chromosomes are the most condensed during metaphase. Okay, so what's the third level of chromosome compaction is this looping and scaffolding, okay? And the scaffolding depends on those non-histone proteins. No histones involved in the scaffolding. Which this is a better way to show it. Okay, so the looping. These proteins, MARs, okay, are going to start looping the DNA. Okay? Remember, this is not naked DNA. This is that 30 nanometer fiber. So there are nucleosomes wrapped around making that solenoid, which are now being looped into what's called this rosette structure. Looks like a flower. Okay. The proteins here also in the center, non-histone proteins, they're going to start scaffolding the rosettes on top of each other, putting them one on top of each other. Okay? So this is the radial loop scaffold model. You got the rosettes being formed, looping, and then the loops are on top of each other, like a giant scaffold. Okay? So that's going to be its most compact state during metaphase. Video.